Today, as I said, the topic is the basis of being different. This title I chose because what I want to emphasize is that even though we all are like same species, we all as individuals are very similar if we look in very gross forms, but there are definite differences among us which make us very different, both in terms of our appearances, both in terms of our susceptibility to various diseases, as well as the behavior. So I won't touch all the aspects, but I will try to give you the genetic background of what makes us different. And ultimately, at the second half of my talk would be how we use this brewer's yeast. Now, yeast has been used since millennium for baking and brewing, and what we have and since early 20th century, it has been one of the most well studied eukaryotic organisms. So, and it was the first eukaryote to be sequenced in 1996. So what we have developed as using yeast is not only a model which is good for studying genetics, molecular biology, cell biology, but also something that allows us to understand understand the fundamental principles which make two yeast strains or yeast species very different. Okay, so this is these are various pictures. Like if you see, first is height. Now height is something very obvious. Like you can see, if I ask all of you people to stand in like ascending order of height, we'll see there's a variation from say 5 feet to 6.5 feet or whatever the range might be. So there is a variation, and it's not that it's not a Mendelian trait. The reason is that it's not an on-off switch. On-off switch would mean that either you have no height or you have full height. Like if you look at Mendel's flower experiments, they were either white in color or red in color or intermediate pink. It wasn't like there was a complete range, whereas in height you will see a complete range from a shortest human being around like 4 feet and the tallest being like, so that's a basketball player in one corner, and that's one of the famous American actors. And same goes for dog variations. Like all the breeds, even though they are all dogs, but they're all different, and each has their specific characteristic. If you look at this is this made to cover of science is like, what is the fruit shape of tomato? Like tomato is one of the model organisms again used for studying natural variation, and you can see these are various tomato species that are commercially grown, and even in India you can find cherry tomatoes which are still, they're Lycopersicon esculentum still, whereas the large hybrid tomato that are usually tasteless but usually used for making curry. So there is a large variation. This is corn, and these are the variants from these are actually commercial variants that are grown across the world. So this is a picture taken from there's a Center for Maize Research in Mexico, and they sort of they have a full variation. Mice is another model system that people use, and this is what's called a heterogeneous stock. They all of the individuals, like the black mouse and the gray mouse, they are sort of the parental strains, and then they cross them to see what variation is there. And they're not only just looking at the fur color which is varying, but they're also looking at various other characteristics. And this is one of the famous projects that is currently on, which is like Genome Project. And the idea behind the project is that we'll sequence 1,000 individuals, human individuals from around the world, and then try to see what is the difference between them, what makes an African, an Asian, a Chinese, or an American. So kind of that way. And this picture is from the 2 strains of yeast that we use. So one is the one on the top is the lab strain, which you don't see, which you see is having an very small region of offshoots, whereas this strain has a large, very extensive projections coming out. Okay, so the basic idea is that to know variation, we have to read the DNA. Now reading DNA has been a slow process initially. Now it has picked up a lot. Like if you see, it took us 10 years almost, or like 6 years to sequence yeast genome, which is like 12 megabase only. It started in 90 and was published in 96. Human took 10 years, and now you are getting a human genome published, like you have Watson's genome, James Watson's genome, Craig Winter's genome, and then there is an African genome, a Chinese genome. So there are like lots of genomes that are coming out because of the changes in the technology. So the basic question is, how many genes are there? What do they do? That is because once you have the genome, you have to understand this. You can get an understanding of that. What are the origins of each of the genes? What makes us human? Now that question is something that concerns the only 1% difference in genome between a chimp and a human. So we are only 99% similar to a chimpanzee's genome, but we are humans and they are chimpanzees. So what makes us human? And within a human population, what is the phenotypic variation? Because as I said, we are all different. It's like it's opposites of hum sab ek hain kind of way. We are all different, and as individuals we have very unique characteristics, and those characteristics, as I said, is because physical characteristics are there, and then the main focus is on the medical side, where we want to study the susceptibility to various diseases. Now, of the diseases that, like many of the Mendelian diseases are known. So like you have a mutation in a mitochondria, you get a very severe mitochondrial defect, and that leads to very short lifespan. Whereas cancer, for example, people know that there are various cancer genes, people have identified various cancer genes, but they have not been able to tell for the fact that having a cancer gene means you have a cancer or not. And even if you have the gene, when would you get the cancer? So it's something to do with not only that particular gene that is required, because that particular gene needs an action of various other genes which are associated with cancers to give a effect. So cancer, type 2, I'll show you a list, extensive list that we have been able to generate. So type 2 diabetes, like people know that diabetes is caused by lack of insulin production, but that's a very simplistic definition of diabetes. Diabetes is not just that if you start taking no sugar means your diabetes would go away. And type 2 diabetes is a very complex phenomenon because you don't really know what exactly causes it. Various neurological disease like Alzheimer's is something that there's a huge variation for. So the main focus of all the studies and the reasons why people want to understand the variation is to understand what is the medical basis or the genetic basis of the differences that we see in the phenotypes of various diseases. Great, thank you. Wow. So here's the list of sequenced genomes, like you see human, like this is around 23,000, that's an estimated number of genes. See, even Arabidopsis has more number of genes than humans. Yeast is 6,000, that's why it's a very successful model, as I'll show you later. So if you see Drosophila, which is a fruit fly, the number of genes is not that large, but the size is a very huge difference over there. So that actually gives us a clue of why humans are more complex than flies. And similarly, but this alone is also not a very clear indication of complexity, because if you look at, can you think of the most, like the organism which would have the largest size of genome? Do you know which one it is? No? No idea? It's amoeba. Amoeba has around 10-fold more size than humans. If you like the chromosomes, if you look at the sequence, like the DNA sequence of a chromosome and add up all the chromosomes. So for example, in yeast, 12 million means 12 megabases. That means the 16 chromosomes that are there in yeast, you count the number of bases in each chromosome and you add total number of bases. So humans have 23 plus the 6 chromosomes, so that's how we can estimate 3,000. Yes, yeah, nucleotides, yeah. Yes, so 3,000 bases. So base pair is the unit of DNA. So each DNA could, it either could be adenine, cytosine, thymine or guanine. So humans have